Okay, so nervous tissue, personal favorite, is really made out of only two ingredients. You have neurons, which are the ones that send the signals, and then you have neuroglia. And so we'll spend a lot of other videos talking about the neurons, how to classify them, their parts, their structures. This one is going to be about those other ones, those support cells, those neuroglial cells. So neuroglia, sometimes known as glia, are going to be separated out depending on where you're at. If you are in the brain or the spinal cord, we call that our central nervous system. So the CNS will have different neuroglia than what we'll have in the peripheral nervous system. So CNS is brain and spinal cord. Peripheral nervous system is outside of that. So we'll call that our PNS. With our CNS, we're going to want to establish the boundary of what is brain and spinal cord versus what's not. And so what we're going to have to do that is our astrocyte. So the astrocyte has a lot of variety in its jobs. One of the key ones that I always like to think of is the blood-brain barrier. And so what you're doing is you're establishing a separation between what is considered the central nervous system and what isn't. And so with an astrocyte, the name tells you what it looks like. Astro means star. Cyte means cell. So it's a cell that's shaped like a star. And so picture like a little octopus with arms going in different directions. And so what they'll do is they'll have some of their little arms and their processes reaching out and touching the blood vessel and wrapping around it. And some will reach out and touch the processes off of the neuron. And so you're also establishing your framework. So it helps hold things in place. They also make scar tissue if there's a damaged area. So they fill in that space to say, oh, don't make wrong connections. Let's just figure out how to work with whatever is left around here. And so astrocytes are going to be more dealing with your structure and regulating your environment. So that's what that blood-brain barrier is doing. It's allowing us for regulation of that environment. We also want to have the ability to insulate the neurons. So remember, with a neuron, you're sending electrical information, and just like electrical information in your home, you need to insulate that. So we're going to have an oligodendrocyte. So the job of an oligodendrocyte is to insulate the electrical signal that's going down the axon, which if you think about it, is like the wire of the cell. So this is going to be insulation. And that insulation is called myelin. So we're going to make a myelin sheath. Myelin is going to be lipid-based, so it'll actually have a white appearance. So when you cut the brain, you have an area that looks gray and an area that looks white. The area that's white is because of the oligodendrocytes. That's the area that sends signal, and that's where the insulation's at. So when we talk about white matter in the brain, we're talking about areas where we have that signal being sent, and that signal is being insulated by this white material made by the oligodendrocyte. Now, oligodendrocytes, when they go to myelinate, they can actually take their process and reach around that axon and curl, curl, curl. So again, think octopus. This one, process is going out, sticking to different things. This one, take one and then curl, and I'm not as flexible, but hopefully you get the idea of where I'm going here. So we're wrapping around and insulating that axon. And so we can wrap around and insulate multiple axons. That's one of the things that's different about the insulator here versus out in the periphery. Here, the brain and spinal cord is only neural tissue. There's nothing else in the way. So we can take that time and that space to curl around a couple of different cells. And so it helps stabilize their positions as well. It'll hold those axons in those places. Being in the brain and spinal cord, one of the things that is unique is we have this blood-brain barrier. So we don't let blood into the brain. We need to create our own transport fluid. We're very, very protective of that central nervous system. So we're going to have our own fluid be made called cerebrospinal fluid. And it's the ependymal cells that are going to do that. And so they'll make your cerebrospinal fluid, or CSF. With CSF, you're making it in spaces that are called ventricles. Those are the spaces, or like when you do the skull, you have like sinuses, it's hollow pouches. Inside of the brain, you have hollow areas that are going to be filled with cerebrospinal fluid called ventricles. So ependymal cells are going to line those ventricles, those spaces, make this fluid. They have cilia on them so they can move that fluid around into circulation. And then also, because we're very protective, we're going to have our own special cleaning crew. And these are going to be our microglia. And so these are going to clean. And they're phagocytes. They go and eat debris, anything that's not supposed to be in the brain or spinal cord. They'll help destroy and remove that. So these are the four helper cells or support cells of our central nervous system. Out in the peripheral nervous system, we'll have something that does a job similar to the astrocyte as far as creating and regulating that environment. And so that's going to be our satellite cell. So this is going to be establishing our environment, which is what the astrocyte is doing. And then we'll also have something that does the job like the oligodendrocyte called a Schwann cell. So these also insulate. Out in the peripheral nervous system, there are other tissues and things to be mindful of. So we're going to be really compact with how we insulate. And so when we go make that myelin sheath, that myelin covering, we're going to take one cell and we're going to wrap all the way around it. And so I think of it kind of like a pig in a blanket. So if you don't know what that is, hot dog with a crescent wrap or a crescent roll, and you wrap it around it and then bake it. Try it if you haven't. That's kind of the look here. So we have our axon, and then we take our cell and we just wrap over and over and over again. And one cell myelinates just one part, and then we'll have another cell wrap, wrap, wrap. There's a little gap in between, another cell wrap, wrap, wrap. And so they're doing the same job as the oligodendrocyte. But you don't have any other neuroglia in the PNS. You don't need to make your own specialized transportation fluid because out in the peripheral nervous system, you already have a transportation fluid. Actually, you have more than one. You have lymph and blood. You also don't need to make your own specialized macrophages because you already have macrophages floating around. You have monocytes and macrophages that can go and eat up debris out in the peripheral nervous system. So these two are very similar to each other. These two you don't need to have a duplicate for because there's already stuff out in the periphery that can do those jobs. So those are your support cells for your neural tissue. Thanks for watching.